Hello everyone, my name is Jyothi Paniyadi and I'm a technical support scientist at ACD. Hope everyone is doing well and staying safe. Today's presentation will focus on how you can incorporate RNAscope in situ hybridization technology in the single-cell RNA sequencing workflow to confirm and spatially map multiple gene signatures. A quick overview of the topics I will be covering today. I will begin by giving a brief introduction to RNAscope technology, followed by a description of our unique probe design and amplification systems that allows for simultaneous detection of multiple genes. We will also discuss the various approaches for analyzing the data that is generated. And lastly, I will briefly touch upon our product offerings that support COVID-19 research, and after which my colleague Jyothi Sheldon will talk in more detail as to how researchers are using RNAscope to map the single-cell transcriptomics information back into the tissue. Standard RNA-seq procedures have an inherent limitation in that the transcriptional signature that we get is only a representative information averaged across heterogeneous cell populations. Gene profiling with single-cell resolution, where we can get cell type specific expression patterns, has therefore gained much ground in recent years. However, single-cell RNA sequencing also has a limitation, and that is lack of spatial context due to dissociation of the cells. Methods like RNA in situ hybridization can help validate these transcriptomics findings and also to study the gene expression in its native environment in the tissue. RNAscope is an ISH technique that is different from other ISH methods because of its unique probe design strategy. It utilizes a unique double Z probe design followed by a signal amplification system that reduces background and improves a signal to noise ratio significantly. In this design, each Z probe has a lower part that is 25 bases in length, and this is complementary to the target RNA, and an upper region which is a 14 base tail sequence that has the preamplifier binding site. The upper and the lower parts of the Z probe are linked together by a spacer sequence. Two such probes must come together to create a 50 base specific binding site that hybridizes with the target region of interest. The informatics algorithm that is used to design these probes ensures that the oligonucleotide sequences are specific and do not cross-hybridize with any other sequence. The top of the Z is the base for the amplification structure. When two Zs hybridize, it creates a binding site upon which a preamplifier can bind and the amplification tree can be built. A standard RNAscope probe for a target sequence of 1,000 bases or more consists of about 20 ZZ pairs pooled together that are designed to hybridize next to each other along a target region. This allows for a tremendous amount of amplification and signal potential. Our ISH technology consists of two unique assays to detect RNA in the tissue context at single molecule sensitivity and with single cell resolution. These are RNAscope and BaseScope. Both these assays are based on the same technology and are capable of single molecule detection, meaning a single dot represents one single RNA molecule or target. RNAscope assay is applicable for mRNA or non-coding RNA species that are greater than 300 nucleotides in length and is available as a singleplex or duplex chromogenic or as a multiplex fluorescent assay. Our V2 multiplex fluorescent assay can detect up to 4 targets simultaneously, while our high-plex fluorescent assay can detect up to 12 targets simultaneously in tissue sections. BaseScope, on the other hand, is a more specialized assay designed for the detection of exon junctions, splice variants, highly homologous or short sequences that are between approximately 50 and 300 nucleotides, and also validated point mutations such as KRAS, etc. It is available as singleplex or duplex chromogenic assay. Here is a brief overview of the RNAscope workflow. RNAscope assay is a slide-based assay, allowing for both chromogenic and fluorescent labeling and is compatible with a wide variety of samples, including FFPE, fixed frozen, and fresh frozen tissues. The assay starts with permeabilizing the sample with reagents provided in the pretreatment kit that enables unmasking of the target mRNAs. This allows the probe to penetrate into the cell and hybridize with the target RNA sequence. The probe is a pool of oligos that are designed using our established bioinformatics algorithm, which allows for target specificity. A series of amplification cascade then amplifies the signal, and RNA molecules are then visualized under the microscope as punctate dots. The signal can also be quantified semi-quantitatively or quantitatively using image analysis software. Here is a schematic showing the hybridization and subsequent amplification of the RNAscope assay. The double ZZ probe will bind to the target mRNA transcript. Once the double ZZ probe binds, it serves as a foundation and binding site for the preamplifier molecule. Once the preamplifier is bound, it serves as a binding site for the amplifier molecules, and each amplifier can further bind multiple labeled probes, sequentially hybridizing to assemble a branching complex at each ZZ binding site. Labeled probes contain an enzyme such as HRP that generates a visible signal after chromogenic reaction, such as with DAB, that is detectable under a standard bright field microscope. The labeled probes can also contain fluorophores that allow for visualization of the signal under a fluorescent microscope. This signal amplification strategy provides for visualization of target RNAs as a single dot, where each dot represents an individual RNA molecule. If a single Z binds to the target mRNA transcript, the preamplifier cannot bind as it needs both ZZs to provide a binding site. This helps to ensure the specificity of the probes and helps reduce potential background staining. This table summarizes all the assays that are currently available from ACD. We offer kits that can detect 1, 2, or more targets at a time. Multiplex fluorescent V2 kit can detect up to 4 targets, and High-plex kit can be used to detect up to 12 targets simultaneously. Let's take a quick look at the assay workflow for V2 and High-plex kits. In the V2 assay, as in other assay types, there are 4 major steps: sample pretreatment, hybridization, amplification, and detection. The channels are developed sequentially after the amplification step, and the TSA-based fluorophores are added separately during the detection steps. This offers the flexibility to the user to opt to visualize any of their targets using the opal dye of their choice. High-plex workflow is performed in 3 rounds, with 4 targets being detected in each round. Round 1 is the longest, taking about 7 to 8 hours, and includes probe hybridization and signal amplification steps. In this round, you will also detect the first set of 4 targets and follow it up with imaging. After round 1 imaging, the cover slips are removed from the slides, the fluorophores that are conjugated to the oligos are cleaved off chemically, and the next set of 4 targets are iteratively detected using the same 4 fluorophore types, followed by imaging. Once all 3 sets of images are generated, they are aligned appropriately using the High-plex image registration software. We provide recommended sample preparation protocols for most of the commonly used sample types. However, if you're working with unique sample types such as whole mount, free-floating, or bone, please feel free to contact our tech support for suggestions and guidance. We have so far talked about how the technology works and the various assay workflows. Now coming to the question of how to analyze the results. Data analysis can either be semi-quantitative or quantitative. For semi-quantitative analysis, we provide some guidelines that I will elaborate upon in a later slide. For quantitative analysis, we generally recommend either ImageJ or Cell Profiler, but you can opt to use any of the other softwares or available free tools that best suit your requirements. We have technical notes and webinars on our website that you can refer to help with data analysis. However, please note that these are simply guidelines, and ultimately researchers must decide how they want to analyze their data and which quantification method they want to use. RNAscope staining is scored by counting the number of dots. Each dot represents 1 molecule of mRNA. Sometimes multiple mRNA copies may show overlapping signals, and this is called a cluster. It is the number of dots in clusters that are important rather than the size or the intensity of the dots. In the semi-quantitative scoring method, the number of dots or clusters per cell is manually counted. With this method, a score of 0 to 4 is assigned based on the average number of dots per cell. We have a guideline document on our website that you can download that describes the different methodologies to quantify your results. If you have further questions, please contact technical support for recommendations. There are many vendors who sell image analysis softwares. There are also some freeware that you can easily download from the internet. Here at ACD, we promote two such popular open source tools for image quantification. One of them is ImageJ, which we used in our in-house projects in the past and have had success. The other is Cell Profiler. We have technical notes and webinars available in our website that provide step-by-step instructions on how to use these tools to quantify your data. On the new product side, we now have a new assay platform called miRNAscope that will be in market soon. miRNAscope is a robust assay offering high sensitivity and high specificity with a universal probe design. The custom probe is designed to detect microRNA, antisense oligonucleotide, or small interfering RNA sequences of interest. The microRNAscope assay can be combined with downstream protein detection and is compatible with FFPE, fresh and fixed frozen, and cultured cell samples. It will initially be available as a single-plex chromogenic assay, both manual and on Leica automation. During these challenging times, I would like to take a few minutes to talk about some of our product offerings that help support COVID-19 research. We have a probe that detects the viral spike protein and another that detects the replicating strand. We also have probes for host cell markers, for example, ACE2, which is an entry receptor for SARS-CoV-2 and has homology with ACE enzyme, which is a key player in the renin-angiotensin system. We have probes for another target that is being studied in COVID-2 research, which is TMPRSS2. This is implicated in the viral and host cell membrane fusion process. Additionally, we also have probes for IL-6, IL-10, etc. to detect the cytokine response during infection. Not listed here but available to order are probes for lung and ARD markers like AQP5, SFTPB, RAGE, KL6, and CC16. And these probes can be used with the red, brown, and duplex chromogenic assays, as well as the multiplex fluorescent V2 assay. For those of you who are just getting started with ACD's assays, we offer a new user program where we pair you up with one of our technical support experts to provide guidance throughout your experiment to ensure a successful run. And for those of you who would like to send in samples and prefer the assays to be run and validated by ACD, we offer academic support services. Our service teams ensures all due care is taken with your samples and that the assay development and analysis are conducted with highest efficiency. With this, I come to the end of my presentation. I will now hand over to our application scientist, Jyothi Sheldon. Thank you for your attention.
Thank you so much, Jyoti, for that great introduction. And now I'd like to go over some of the applications of RNAscope technology in the field of single cell. The RNAscope technology has been widely applied for numerous technical and research applications for both academic institutions and biopharma companies. Today I will be focusing on the validation and spatial mapping of high throughput data, namely single cell RNA sequencing data with RNAscope assay. Complex, highly heterogeneous tissues such as the brain and tumor, as well as processes such as embryonic development, are comprised of multiple cell types and states. Precise transcriptomic characterization of these tissues can enable identification of new cell types, predictive biomarkers, new therapeutic strategies, and more. However, interrogation of these complex heterogeneous tissues requires a highly specific, sensitive, and multiplexed spatial approach with single-cell resolution. Traditional transcriptomic analyses have relied on bulk tissue analyses, such as PCR, which provide an average level of gene expression in a sample but loses the spatial and single-cell information. This is akin to a fruit smoothie, which can be a delicious drink, but you can only guess what is in the smoothie. The advent of single-cell transcriptomic analysis, such as single-cell RNA sequencing, have now been able to provide molecular information at the single-cell level, and, to come back to the fruit analogy, can now provide you the menu and tell you what fruit and how much of each fruit went into that smoothie. However, these techniques still don't provide you with how those cells are organized in the tissue context, and this is where spatial analysis come into play. Single-cell multiplexing spatial analysis that detect RNA can provide transcriptomic information with spatial organization while retaining single-cell resolution. So now, instead of an average blended smoothie with mystery ingredients, you can see all the fruit and how it is spatially arranged. Current single-cell transcriptome studies utilize dissociated cells and result in the loss of spatial organization of the cell population in the tissue context. Validation and spatial mapping of single-cell analysis can be obtained using assays that retain spatial organization, such as the RNAscope in situ hybridization assay. The multiplexing capabilities of the RNAscope technology, coupled with its high signal-to-noise ratio, enable single-cell and single-molecule detection of transcripts in the tissue context, providing confirmation of high-throughput single-cell transcriptomic results, as well as adding spatial information to these datasets. Applications of incorporating the RNAscope technology into single-cell sequencing workflows include spatially mapping a cell atlas, visualizing gene signatures of newly identified cell types, classifying highly heterogeneous cell types, confirming new therapeutic cell types, characterizing immune landscapes, identifying new immunotherapy targets, analyzing or predicting response to drug treatments, confirming publicly available datasets such as the TCGA and Tabula Muris. I'd first like to show some examples of how RNAscope was used to complement cell atlases generated by single-cell RNA sequencing. Vagal sensory neurons are necessary for the control of organ functions such as cardiopulmonary and gastrointestinal systems. But knowledge on the complexity of neuron types involved is missing. Researchers from Patrick Ernfor's lab at the Karolinska Institute present a comprehensive identification, classification, and validation of the neuron types in the jugular and nodose-derived vagal ganglia. 25 RNAscope probes were used to not only validate neuronal types identified, but also to quantify the relative proportions of these neurons. Jyothi shows some representative data interrogating the nodose vagal neurons. The Human Cell Atlas, led by Sarah Teichmann, published this seminal paper in Nature Medicine, describing a cell atlas of the human healthy lung, as well as the asthmatic lung. With regards to asthma, using more than 25,000 cells from healthy and asthmatic lung, they revealed mucus cell hyperplasia in the asthmatic lung. The multiplexing capabilities of RNAscope fluorescent assay allowed for confirmation of a strong increase in mucus-ciliated cells in the asthmatic lung by detecting co-expression of the MUC5AC, a secretory marker, and FOXJ1, a ciliated cell marker in the same cells, as indicated by the white arrows in the image on the bottom right. Human epidermis is a complex organ and has multiple lineage cells that must coordinate with the immune and nervous systems. In this paper by Jeffrey Cheng, scientists at UCSF Department of Dermatology report single-cell RNA sequencing data of over 92,000 normal and inflamed human epidermis samples, revealing a discrete set of specialized keratinocytes that exhibit a distinct composition at different anatomic sites. They used RNAscope multiplex assay to validate their single-cell RNA sequencing data by looking at gene expression in differentiating scalp keratinocytes and identification of markers for specialized hair follicle subpopulations. The cell atlas initiatives in the normal and disease states have now empowered researchers to combine single-cell analysis with spatial analysis for various pathologies. Last year in Nature, 2 studies profiling the single-cell transcriptome of the mouse and human airway epithelium both serendipitously identified a new cell type that could be linked to cystic fibrosis. While generating cell atlases of the mouse and human lungs, these authors also identified a new airway epithelial cell type that expresses both FOXOT1 and CFTR, a gene that is mutated in cystic fibrosis patients and responsible for transporting chloride ions out of the cell, a process that is impaired in cystic fibrosis patients and leads to mucus covering the cells. They termed this FOXI1 and CFTR dual positive cells pulmonary ionocytes because they seem to be involved in transporting ions across lung tissue. Molecularly characterizing these new cell types could aid in identifying the appropriate cellular target for cystic fibrosis gene therapy. In this study from the Broad and MGH, the authors generated a single-cell atlas of mouse tracheal epithelium, in which they identified functional variations in club cells based on their location, a distinct cell type in high-turnover squamous epithelial structures that they termed hillocks, and disease-relevant subsets of tuft and goblet cells. They also discovered a rare cell type, the FOXI1 positive pulmonary ionocyte, that is also the major source of CFTR in both mucus and human airway epithelium. They then went on to show that knockout of FOXI1 in mouse ionocytes caused loss of CFTR expression and disrupted airway fluid and mucus physiology, phenotypes that are characteristic of cystic fibrosis. The RNAscope multiplex fluorescent assay validated single-cell RNA sequencing data and identified the rare FOXI1 positive and CFTR positive cells in human bronchi. In this study from Novartis and Harvard, researchers performed single-cell profiling of human bronchial epithelial cells and mouse tracheal epithelial cells to obtain an unbiased, comprehensive census of cell types in the conducting airway. Unexpectedly, they also identified the same novel rare cell type they called the pulmonary ionocytes that co-express FOXI1 and CFTR, the gene mutated in cystic fibrosis. In this study, RNAscope was used to validate that both mouse and human ionocytes co-express FOXI1 and CFTR. In addition, the assay demonstrated that CFTR was highly enriched in FOXI1 positive cells, but not FOXJ1 positive cells. They went on to show that notch signaling is necessary, and FOXI1 expression is sufficient to drive the production of the pulmonary ionocyte, and that the pulmonary ionocyte is a major source of CFTR activity in the conducting airway epithelium, which could better inform therapeutics for cystic fibrosis. Transcriptomic profiling of complex tissues by single-nucleus RNA sequencing has also been gaining traction. Alzheimer's disease, or AD, is an age-related neurodegenerative brain disorder of which the molecular and cellular complexity remains poorly understood. MIT researchers analyzed over 80,000 single-nucleus transcriptomes from the prefrontal cortex of 48 individuals with varying degrees of AD pathology, providing a first blueprint for interrogating the molecular underpinnings and cellular basis of Alzheimer's disease. RNAscope duplex assay confirmed cell type specific downregulation of differentially expressed genes, revealing significantly fewer excitatory neurons marked by SLC17A7 with detectable NTNG1 expression in brain sections from AD patients compared to the no pathology patients. In this Nature paper, the author Sarah Jekyll identified through single-nucleus RNA sequencing immune oligodendroglial cells, a population that expressed both canonical oligodendrocyte genes as well as genes associated with microglia, hence why they were named oligodendroglia. Interestingly, these immune oligodendroglia are enriched in the human multiple sclerosis brain. Dual BaseScope ISH-IHC revealed expression of microglia markers CD74, in olig-1,2 positive oligodendrocytes in an MS lesion, confirming the presence of this hybrid cell type in the human MS brain. While the brain and many other organs have been analyzed by single-cell techniques in numerous studies, the highly heterogeneous tumor and its microenvironment is quickly becoming a top tissue sample to examine at the single-cell level. So let's take a look at some examples of single-cell RNA sequencing in tumor biology. Let's start with this one. This is a great example of a paper from Broad Institute where researchers dissected the cell types in IDH mutant gliomas by combining single-cell RNA sequencing profiles of 14,226 cells from 16 patients with bulk RNA sequencing profiles from 165 glioma patients. In this paper, they found an increase in macrophage over microglia expression programs in the TME as the tumor grade progresses. RNAscope in situ hybridization assay validated the single-cell RNA sequencing results and demonstrated that inter-tumor variability in microglia macrophage states correlated with tumor grade, showing that cells from a high-grade tumor were preferentially associated with macrophage-like expression states. For instance, the tumor MGH56 is a low-grade tumor and has few microglia and no macrophages, whereas tumor MGH42 is a grade 4 glioma and contains only macrophages. However, RNAscope also revealed that tumor MGH43, an intermediate grade 3 tumor, contains cells co-expressing the microglia marker CX3CR1 and also the macrophage marker CD163. By shedding light on the cellular composition of IDH mutant gliomas, this data could aid in the design of immunotherapies targeting gliomas. This is another great example from Nature Communications. Cancer-associated fibroblasts, or CAFs, are a major constituent of the tumor microenvironment in many of cancers. However, morphology remains the most common way to distinguish CAFs as molecular markers and are neither all-encompassing nor specific. This suggests that CAF may be a diverse group of cells made up of several subtypes. Bartoshek utilized single-cell RNA sequencing to delineate the heterogeneity of 768 CAFs isolated from the genetically engineered MMTV PYMT mouse model of breast cancer. They defined 4 transcriptionally diverse subpopulations of CAFs, ones with vascular gene signatures, ones with extracellular matrix gene signature, one with cell cycle gene signature, and one with developmental gene signature. Each of these subpopulations was discriminated by different gene expression programs representing different functionality and unique spatial location within the tumor parenchyma and as identified by the RNAscope assay. For example, using in situ hybridization for the VCAF mRNA markers Notch3 and KCNJ8 in combination with immunofluorescence for the vascular protein marker NID2, it revealed the vascular CAFs are localized predominantly near the vasculature. Furthermore, CAF populations are distinct with no overlap in signal between extracellular matrix CF and vascular CF. This study reveals a previously unappreciated functional diversity within the tumor microenvironment that can aid in further development of tools for precision medicine. Similar group of scientists also showed that glioblastoma has different subtypes that exhibit different molecular states. They identified state-specific molecular markers and visualized these subtypes using RNAscope. They also identified that tumors can have more than one states called hybrid tumors, and these hybrid cells can also be visualized since they express both the molecular markers. Scientists from Harvard used RNAscope duplex assay that allowed the visualization of the different cellular states and their distribution in human glioblastoma patient samples. In order to provide targeted therapy with the highest efficacy, it is important to completely characterize the cellular composition of medulloblastoma tumors. Scientists from Mass General Hospital Department of Pathology performed single-cell RNA sequencing to define the cellular states underlying each of the 4 different medulloblastoma subgroups. RNAscope duplex assay was used to visualize different metaprograms within the Wnt subgroup of medulloblastoma patient tumors. The last research area I'd like to highlight is single-cell spatial analysis for stem cells and development. During homeostasis, LGR5-positive crypt cells drive turnover of the intestinal epithelium. However, even though LGR5-positive cells are lost during injury, the epithelium can still recover. Therefore, an additional population of quiescent stem cells must exist to drive epithelial renewal in response to injury. Researchers from Alex Gregorius' lab at Mount Sinai Hospital in Toronto used single-cell RNA sequencing to profile the regenerating mouse intestine and identify a novel stem cell population, termed revival stem cells, or REV-SCs, and shown as cluster SSC2. They found these stem cells were enriched for genes involved in cell survival, such as clusterin or CLU. During homeostasis, CLU-expressing cells are rare and distinct from LGR5-positive cells, and in response to irradiation, the number of CLU-positive epithelial cells expands. The RNAscope multiplex assay revealed that rare CLU-positive cells in the normal intestinal crypt are distinct from LGR5-positive cells, and that there is a significant increase in CLU-positive cells in the damaged intestinal crypt. Furthermore, the researchers also identified YAP1, a transcriptional regulator, as important for regeneration of the intestinal epithelium. During intestinal regeneration after injury, YAP1 induces reversal stem cells that will create new LGR5-positive cells and thereby restore the epithelium. Single-cell RNA sequencing revealed that the YAP1 gene signature was present exclusively in the SSC2 cluster, and deletion of YAP1 in the intestinal epithelium suppressed emergence of endogenous CLU-positive cells, as demonstrated by dual RNAscope ISH immunofluorescence technique. Organogenesis requires the complex interactions of multiple cell lineages that coordinate their expansion, differentiation, and maturation over time. Pancreatic organogenesis is a complex and dynamic process that ultimately results in the generation of multiple cell lineages that perform the functions of the mature organ, including regulation of glucose homeostasis by the endocrine compartment and production of digestive enzymes by the exocrine compartment. Researchers from Julie Sneddon's lab at UCSF profiled the cell types within the epithelial and mesenchymal compartments of the murine pancreas across development using a combination of single-cell RNA sequencing, immunofluorescence, RNA in situ hybridization, and genetic lineage tracing techniques. Among several major findings, they identified a previously underappreciated cellular heterogeneity of the developing mesenchyme, as shown here in E14.5 pancreas, and confirmed by dual RNAscope ISH-IF technique. This work establishes a roadmap of pancreatic development and demonstrates the broad utility of this approach for understanding lineage dynamics in developing organs. A great way to help further characterize cellular gene expression is to combine ISH with IHC on the same slide to detect simultaneously RNA and protein. And we've seen some examples of these on multiple publications described earlier. What we refer to as dual ISH-IHC can be used to characterize cell type specific expression, identify origin of secreted proteins, visualize cell markers with RNA of interest, visualize RNA binding proteins and their target RNA, and dissect regulation of gene expression. Because of the similar workflows between ISH and IHC, including sample fixation, pre-treatment, probe hybridization, signal detection, and data analysis, as well as the unique benefits of each assay described earlier, ISH and IHC are ideal to combine into one workflow in which the RNAscope ISH assay is performed first, followed by the IHC assay. We do have some recommendations to ensure success with your dual ISH-IHC assay. First, all dual ISH-IHC protocols require optimization. In general, it is recommended to combine a working RNAscope protocol with a working IHC protocol. In addition, work with antibodies and a protocol that are known and already established with your tissue samples. Second, it is advisable to perform ISH first, followed by IHC. And third, it is advisable to optimize the IHC assay separately using the RNAscope pre-treatment reagents to ensure your protein can still be detected following RNAscope pre-treatment. Lastly, the dual ISH-IHC assay works better for highly expressed proteins due to protease treatment that is used during the ISH protocol. So let's see some of the examples where researchers have used RNAscope in this dual ISH-IHC or IF technique. Hepatic stellate cells, HSCs, are a major contributor to liver fibrosis, but were previously thought to be homogeneous. Using single-cell RNA sequencing, Dobie revealed two subpopulations of HSCs with a unique gene signature, the ones that are associated with portal vein and the ones associated with central vein. Using RNAscope in situ hybridization and immunofluorescence, they reveal zonation of the HSC subpopulations across the healthy liver. In addition, it also validated the markers associated with these individual HSC subpopulations. Here is another technique that allows for characterization of complex tissues and spatial profiling at a single-cell resolution. The image is a representation of the joint workflow between the RNAscope assay and the NanoString Genomics DSP platform. The process starts with identifying the targets using the RNAscope probes that will molecularly guide to your region of interest, followed by hybridization of the Genomics DSP probes and profiling of the region of interest. The quantification is done using the NanoString encounter assay. At the end of this run, the targets of interest can be further validated using the RNAscope assay on a serial section at a single-cell resolution. Genomics DSP and RNAscope are individually very powerful techniques, but with the joint workflow, we can have spatial gene expression profile with high plexing capability while retaining the morphological context, which is extremely significant for heterogeneous tissues like tumors. With the joint workflow, we have combined the best capabilities of both technologies while maintaining the technical feasibility for each assay. I would now like to give an introduction to our high-plex assay. Jyoti did a great job earlier of highlighting our various products and their workflows. I'd like to highlight and go over the applications and utilization of the high-plex assay in single-cell RNA sequencing workflows. To fully characterize and spatially map the diverse cell types identified by single-cell transcriptomics, RNA-based in situ hybridization assays capable of higher multiplexing are critical. ACD's RNAscope high-plex assay allows researchers to visualize up to 12 targets on the same tissue section at a single-cell resolution. With the multiplexing capabilities of this assay, researchers can A. find your population of cells by using a cell-specific marker. They can confirm your single-cell RNA sequencing results by simultaneous detection of additional gene expression target markers. Or C. They can characterize complex heterogeneous tissues by mapping up to 12 targets simultaneously in the tissue context and identifying multiple cell subtypes. What you see on the screen is the workflow for performing the high-plex assay. I'm not going to go on it in too much detail as Jyothi's already talked about it in her overview. The idea is you can do up to 12 mRNA targets on a single tissue section. The way it works is you do 4 targets in 3 iterative rounds, and at the end of the 3 rounds, you combine your data together using the high-plex image registration software. So let's look at an example of the internal applications data to show the utilization of this assay. In this paper by Goxie, published in Cell Reports in 2016, researchers at Stanford identified novel subtypes of the D1 and D2 striatal medium spiny neurons by analyzing over 1,200 striatal cells using single-cell RNA sequencing. They identified discrete cell types that exist in a continuous spectrum of transcriptional states and found that functional diversity within a complex tissue arises from a small number of discrete cell types. However, they did not confirm their single-cell RNA results, nor did they show the spatial relationship between the D1 and D2 subtypes. Therefore, we employed the RNAscope technology to confirm these results and spatially map the subtypes in the mouse brain. and I will show you some example of that data. So on the screen, you'll see the experimental panels that were run using the HyPlex assay. Because the D1 and D2 MSN subtypes were each characterized by 8 genes, the RNAscope HyPlex 8 reagent kit, which detects 8 targets simultaneously and was a perfect assay to interrogate these cell types, was used. Furthermore, we could spatially resolve both D1 and D2 MSN subtypes simultaneously using the RNAscope HyPlex 12 reagent kit, which detects 12 targets. Each panel was run individually on one sagittal mouse brain tissue section, allowing us to visualize multiple targets while preserving the number of samples tested. On this slide, you can see we've registered the RNAscope 8-plex assay, where we've identified the major and the minor D1 subpopulations. The inset shows populations of MACE2 high D1 and D near high D1 cell types that were identified by single-cell RNA sequencing. Similarly, for the D2 MSNs, we detected both the major and the minor D2 subpopulations, as seen in the insets by CalB1 high and CART-PT high. Having the ability to test all these probe combinations on the same slide not only limits the use of the tissues, but also provides valuable spatial information of these gene sets on the same plane that can be lost by performing multiple runs on separate sections. The researchers at Stanford also highlighted an important finding in which they identified a group of cells that expressed both the DRD1A and DRD2 genes. To resolve further heterogeneity between the two MSN subtypes, we performed the RNAscope HyPlex assay with 12 targets. The following slide was created using visual tools to more clearly visualize and highlight the data from this experiment. This slide shows data from our RNAscope HyPlex assay with 12 targets, where we compile the images using a HyPlex image registration software. With this assay, we could visualize the heterogeneity of these cell populations on the same tissue section. This looping image was created using online tools to more clearly visualize and highlight the data from this experiment. By doing so, you can clearly see which cells are expressing the various combination of gene targets. This is the still image from the previous video where we have simultaneously detected both the D1 and the D2 striatal MSNs. The researchers at Stanford also highlighted an important finding in which they identified a group of cells that expressed both the DRD1A and DRD2 genes. With the use of our HyPlex registration software, we now have the capability to visualize these specific cells expressing targets of interest or refine gene signatures by toggling on and off different channels. By selecting for our two primary genes, we were able to detect the novel cells expressing both of these genes as highlighted in the publication. The growth and adoption of the RNAscope technology is best exemplified by the number of peer-reviewed publications. We had our first publication in 2011, and since then, there have been over 2,700 peer-reviewed papers published using the RNAscope technology in numerous journals, including many of the top-tier ones. The RNAscope technology is highly relevant across multiple fields of research, with top areas being neuroscience, cancer, infectious diseases, and etc. Furthermore, the incorporation of the RNAscope technology into single-cell RNA sequencing workflows is demonstrated through the rapid growth of publications using the assay to confirm and spatially map single-cell RNA sequencing data in the tissue context, with over 80% of these single-cell papers in very high-impact journals, including Nature, Cell, and Science magazines. So in summary, RNAscope uses a unique probe design strategy to dramatically improve signal-to-noise ratio. We currently have over 15,000 probes on catalog, but new probes can be designed and made within 2 weeks for any target and any species. RNAscope provides both quantitative and spatial information on your targets in the complex tissue environment at a single-cell resolution. The newest assay, BaseScope, can detect splicing variants and short sequences down to 50 nucleotides. RNAscope assays can be complemented by immunohistochemistry or immunofluorescence for simultaneous RNA and protein detection on the same slide. We have lots of new webinars coming up For registration information or any other information related to the RNAscope technology, please visit us on the webs at acdbio.com or contact us at the email addresses on the screen. Thank you so much for your time, and I hope you stay all safe and healthy. Thank you.